Namaskar, my dear friend. You are again welcome to my YouTube channel, DP Forensic Lab. I am DP Gangwar, Director of DP Forensic Lab, former Assistant Director, HOD Physics and Cyber CFSL Chandigarh. In today's video, I will discuss about techniques and standard used in forensic analysis of the cell samples. As we know that we frequently require to compare the two cell samples. One samples collected from the scene of crime, maybe from the cloth of the victim or any other source like a tires, etc. And then one sample collected from a suspected place to find whether these two cell samples are the similar or the coming from the same source. So we can establish the presence of a person or the victim are connected from the scene of crime. So in many cases, it is become very important to compare the Y samples. So in today's video, I brought the standard or the techniques used for comparison of the cell samples. This type of information may be very useful for the professional who are just interning in the field of the forensic science. So this may be very, very useful. So the first standard used for the forensic comparison I have brought from the DFS MHA, Government of India, New Delhi. They have some manual which are available on the net, open source. They have mentioned the techniques, visual examination of the cell samples for determination of the various features like some foreign materials or unwanted materials or any other things are present in the both samples or not. So many things. And other is a microscopic examination. Both samples should be examined under the microscope, under microscope for determination of the various features or the parameters. Another is a particle size analysis. The size of the particles in the cell samples, if the both cell samples are the same or the similar, the particle size or the constitute of the soil to be similar. So this technique used for the particle size analysis. Another is the ignition test. The sample is heated, ignited at a particular temperature, and this loss is measured. If the both cell samples are the similar, they contains the similar organic contents, the loss of ignition should be same or the similar. Another is the density gradient analysis. If the cell sample are similar, the sample should contain the density in the similar. So this technique is used. This is a very sensitive technique. Yeah. And another is the pH meter measurement. pH value, we should measure the both soil samples. So these are the techniques used for the forensic comparison of the cell samples. I will try to make the separate video on the each techniques. Visual examination, microscope examination, especially particle size analysis of the cell samples, ignition loss, density gradient, or this pH meter. I will try to make the separate video on these different techniques. And other important techniques or the manuals I brought from the ENFSI, European Network of Forensic Science Institute. They prepared a manual procedure. This is a source year of this, and they have mentioned the example of the analytical techniques which can be considered for comparison of the cell samples as per the ENFSI standard. So here, this is general characteristic. General characteristic stereo microscopy can be used. Color of the soil using the spectrophotometer. Measurement of the pH values. Organic matter contents examination. Can be used the loss of ignition or particle size distribution. A general characteristic. And inorganic constituent can also be analyzed using this type of the techniques: XRD, XRF, spectrometry, ICP-MS, ICP-OES, microscopic analysis for the heavy minerals, and SEM-EDX, scanning electron microscope energy dispersive analyzer. So these instruments may be used for the inorganic component. And for the dead organic component may be present in the cell samples. This may be microscopic analysis for a plant remains like a pollen, diatom, etc. Gas chromatography for the plant wax or the markers, etc. for the determination of the dead organic component present in the cell samples for the comparison of the two cell samples. Now organic, living organic component, a non-human DNA may be present, diatoms, mites, etc. So this can be used as per the requirement. So I have brought two standard, DFSS standard and ENSS standard for comparison of the cell samples. So these two standard I think is sufficient for comparison of the cell samples as per your requirement or the facility. You can choose the on standard. Now here are the two important cases I want to discuss about. One case is that we have identified the path of the terrorist walking nearby the river. The sample from the shoes or the cloth collected, and then the sample from the different place here, or the other suspect place collected as a control sample, and we analyze, and we found that the question sample found on the shoes or the cloth was similar this side. So I have identified the path of the terrorist walking. Another important case, a dead body found in the farmhouse in a paddy seeds. So the soil collected from the body, clothes, and the other sample from the control sample from the field. In this case, beside the soil examination, we found some paddy seeds was present in question sample as well as the control sample. Paddy seed. So in the first case in my life, I examined the paddy seeds also. So for the comparison of the soil samples. So in this video, I have tried to brought a brief to describe the possible techniques can be used for the forensic comparison of the soil samples. I will make the separate video of the separate techniques. Thank you, dear friend. If you think this video is some informative, please like, subscribe my channel, and also give the comment. Or for any further help, even the training or anything more, you can contact me at this address. Thank you, dear friends.